We have studied in our previous classes about the flower. What is a flower? What is a complete flower? What is the function of a flower? What are accessory walls? What are productive walls? Today we will discuss development of male gametophyte. What is male gametophyte? And what is female gametophyte? So, to know that, I just recall you the essential features of sexual reproduction, which we have studied in previous chapter. That alteration of generation is necessary in sexual reproduction. What is alteration of generation? Alteration of generation means male, female who are diploid will first produce germ cells, sex cells which are haploid. So this process is called gametogenesis. And when fusion of this gamete occurs, again a first cell of life zygote comes in appearance. So from diploid, haploid generation came in origin, which forms again the diploid zygote. So in the same way, in plants also, which is diploid, flowering plants are diploid, the vegetative life cycle contains two sets of genome in their cells. So we do not use the term diploid for plants, we use sporophyte for that, sporophyte. And for haploid generation we use gametophyte. So it is also necessary in plant for sexual reproduction formation of gamete. So that generation of life cycle in plant which produces gamete are called gametophytic generation. So today in this class we will see the development of male gametophyte. We have studied flower. What is the male wall of a flower? We all know that is androecium. Androecium. And androecium is made up of stamen. The individual unit of androecium is stamen. Stamen is made up of two parts. Now, it is anther, and this one is called filament, which joins it with the floral base, and it is known as filament or stalk. So when we will get the transverse section of the anther, vertical section of the anther, we will see, we can see it with our eyes that it is divided into two parts. It is dithecous, two theca, dithecous or bithecous, and each theca has two lobes. When we will get the vertical section, you know, in biology everyone know how do we prepare sections with blade or razor. So when we will make the section, then we will find that each theca there is no any line but to make you understand, I just show you that each section contains two lobes, which is called sporangium, and each sporangium is surrounded by four layers of cells, four different layers of cells. The first layer of the cell is called epidermis. The second layer is called endothelium. The third layer, in which there are 3 to 4 layers of cells which are very loosely arranged, are called middle layer. And the innermost layer which provide nourishment to the sporogenous cells present inside sporangium are called tectum. So, how this structure forms from simple meristematic cells in bird stage is a topic of developmental or morphological biology. We will consider here the sporogenous cells, which differentiates into pollen grains, are known as pollen mother cells, PMC. They get separated from the tissues around them by some kind of secretions, and they become separated from the mother tissue in this lobe, and this lobe is known as microsporangium, as it will hold or it will prepare microspores. So PMC stands here for pollen mother cells. Pollen mother cells. So pollen mother cells go for meiotic division and forms pollen grains. A cell goes for pollen mother cell goes for meiotic division. This is 2n, and this will produces 4 cells. This produces 4 cells which are called pollen grains, but they will not be separated from their origin. Earlier, when the meiotic division occurs, they remain present united either in the form of tetrahedral structure or cylindrical structure, whatever may be the shape. When the 4 daughter cells remain united inside the microsporangium, such structures are called pollen tetrads. After some time they become separated and they are known as pollen grains. Pollen grains. So, one thing here we have studied that Ambrosium is the male wall, which is made up of stamen. A stamen has 2 parts, filament and anther. A matured anther has 4 microsporangium, in which microspores or pollen grains develop by the process of cell division, that is meiosis. Earlier it occurs in the form of tetrad, then it gets separated and known as pollen grains. Now we see the structure of a pollen grain. sporogenous cells or PMC surrounded by septum divide meiotically to form haploid pollen grains. Pollen grains are known as male gametophytes. Now, the male gametophytes or pollen grains is made up of two layers. That means they are covered by two layers. Outer layer is exine and the inner layer is intine. Exine is the hardest known biological material, whereas intine is similar to cellulosic compounds. Exine cannot be broken biologically, but this structure contains a place where exine is missing. Such a space is called germ pore. Germ pore is the place from where the pollen tube germinates, forms during the process of siphonogamy or fertilization in the flower. Because exine cannot be diluted by enzymes present in living world, that is why there is a pore from where pollen tube can form. We know that we can get anthers in fossils form in coal blocks. How they survived so long period? Due to exine. So it is also known as sporopollenin. Exine is also known as sporopollenin. Now, when the pollen grain inside pollen grain, the nucleus divide mitotically in two parts. Mitotically they divide and form two cells inside there. That is called the bigger part form vegetative cell and the nucleus is called vegetative nucleus, and the smaller part form generative cells or the nucleus is called generative nucleus. This is called two-celled stage of pollen grain. Most of the angiospermic plant, in most of the angiospermic plant, pollen grain get released at this space. two cell stage. And when the pollen grain gets released, we know till that time only two layers are present, and remaining two layers, middle layer and septums, get diluted in the nourishment of pollen mother cells and meiotic cells, which involves in the process of meiotic cell division or in the process of formation of pollen grains. So when the anthers mature, till that time two layers are left only. The outer layer is epidermis. Epidermis is made up of dead cells also. Only endothecium and epidermis is the layer which are present at the time of maturity. Rest of the layers get dissolved. So when the anther matures, in most of the angiosperm, the pollen grains get released at two cell stage. But in the rest, the generative nucleus divides into two main nuclei, and this is called three cell stage. Rest of the angiosperm releases at three cell stage. This pollen grain is also known as sporoderma. It contains two male nuclei. In angiosperm, double fertilization occurs. The one male nuclei fuses with the female gametophyte, contains egg. The one male nuclei will fuse with that egg, and the other male nuclei will fuse a diploid structure inside embryo sac, which is known as polar nuclei, and they will form a triploid structure by fusion, that is called primary endosperm nucleus. After the formation of zygote and primary endosperm nucleus, zygote will remain in rest for a certain period. Till then, primary endosperm nucleus will divide rapidly and will make the entire embryo sac full with cells of them, and they are known as endosperm. Endosperm provides nutrition to the developing embryo. So we will see that all these things in next topic. Here we conclude our class by pointing you that pollen grain is the male gametophyte. That means the first cell which contains haploid nucleus. Then this generation contains two male nuclei which are haploid in nature and are the male gametes for the process of sexual reproduction. So in this class we have studied how the development of male gamete, how and where the development of male gametophyte occurs. So what are the important points? I just focus upon that. Androecium is the male part of a flower, male word of a flower. Androecium is made up of individual unit known as stamen. A stamen has two parts, filament and anther. Anther has two parts from outside, and each part contains two lobes. Then there are four lobes in an anther, and in the middle of that the sporangium is present. That means sporangium is four sporangia inside anther is surrounded by four different layers of cells. The outermost layer is called epidermis. Then after epidermis is endothecium. After endothecium there are 2 to 3 layers of loosely arranged cells known as middle layer. Then the innermost layer around the sporangium, microsporangium, is septum. When the anthers mature, septum and middle layers get degenerated due to the function they do of nourishment for the sporogenous cells. Now the sporogenous cells or pollen mother cells go for meiotic division and they form pollen tetrad. Pollen tetrads are the 4 cells united in which 4 cells are haploid and are produced by the process of meiotic cell division. When they get separated, they are known as pollen grains or sporoderma. They have 2 layers of cell wall. One is exine and the other is intine. Exine is the hardest known biological material. Intine is the substance similar to cellulosic compound. And there is a place on the surface where exine is absent, that is called germ pore, from where the pollen tube forms. We get pollen grains as fossils in coal block just because of sporopollen and the exine. Most of the angiosperm get released at 2-celled stage and the rest of them releases at 3-celled stage. The pollen grain is also known as sporoderma. So we will study female gametophyte development in our next class. Till then, goodbye, stay at home, stay safe. Bye.